Good afternoon, everyone. We're going to get started here in just a moment. Just want to give folks a couple of seconds here to get logged in to the webinar, but we'll get going in the next couple, 10, 15 seconds. All right, looks like people are making their way in. So we're going to get started today. So thank you everyone for joining PPMD this afternoon for this webinar with Dyne Therapeutics. I'm Eric Camino. I'm PPMD's Vice President of Research and Clinical Innovation. I'm going to be joined for this webinar by Ash Dugar, the Chief Medical Affairs Officer for Dyne, and Catherine Beaverson, the VP of Global Patient Advocacy. And they're joining us today to provide some clinical updates from DELIVER, which is their phase 1/2 study of DYNE-251, which is a next generation exon skipping therapy for patients amenable to exon 51 skipping. This webinar is being recorded, so if you have any friends or family who would find this relevant, it'll be up on the on PPMD's website in the next week, so feel free to share. And then as we go through the webinar today, if you have any questions related to what we're discussing during the presentation around Dyne's platform, exon skipping, please feel free to put those questions into the chat feature at the bottom of your Zoom, and we will try to get to as many of those questions in the open Q&A as we can. So I think with that, we can get started, and I'm going to hand it over to Ash for this first portion of the webinar.
Great, thank you, Eric. As Eric mentioned, my name is Ash Dugar. I'm the Chief Medical Affairs Officer at Dyne Therapeutics. And first, I'd like to thank Emily, Eric, Pat, and the entire PPMD organization for the opportunity to present our latest data release from May from our Duchenne program. It's always a privilege to speak to this community, and I want to thank you for your time and attention today. Also, as Eric mentioned, with us today is the head of patient advocacy at Dyne, Catherine Beaverson. And I will take a second to gush about Catherine. As many of you know, she has been part of the neuromuscular advocacy community for over a decade, trained geneticist, and brings a tremendous amount of experience and passion to this role. And so I'm really fortunate that she's here with us today. And Catherine will be joining at the end of the talk for questions as well. Next slide, please. I'd like to remind everyone that we'll be making forward-looking statements today, and you can refer to our website should you want more information. Next slide, please. So you've seen me present this slide in the past, and it's really important to us. At Dyne, it all starts with our mission, which is to deliver life-transforming therapies for people living with serious muscle diseases. And this mission is woven deeply into our core values, and it inspires us, our employees, each and every day. Next slide, please. You can go ahead and build it, please. So I know this, this community. I know all of you are listening and have done a tremendous amount of work to understand the disease biology, the modalities that have been tested for Duchenne. And so I won't spend a lot of time on some of the limitations and challenges. So I'll just hit a few highlights because you are so knowledgeable in this space. At a high level, there are three key challenges as to why currently available PMOs, or phosphorodiamidate morpholino oligomers, that they are a type of antisense oligonucleotides used in Duchenne. There are three key challenges as to why those currently available PMOs have significant limitations on dystrophin restoration and ultimately functional improvement. The first challenge has been the ability to get to the nucleus. So that nuclear localization of these PMOs, or antisense oligonucleotides, to be able to harness the cellular machinery to correct the genetic problems. The second challenge has been getting targeted delivery to muscle fibers. Historically, it's been very tough to be able to get enough of the PMO into the most important muscles and tissues, such as the skeletal muscle, including diaphragm, cardiac muscle, as well as other muscles like smooth muscle. The third is broad distribution. So getting in those muscle cells broadly so that we are able to actually have a change that is going to be global in nature. And so really being able to have broad distribution in the muscles that matter for individuals suffering with Duchenne. Next slide, please. So at Dyne, the FORCE platform was developed to try and overcome these challenges. The platform is expressly designed to efficiently and reliably deliver antisense oligonucleotides like the PMOs to skeletal muscle, cardiac, and smooth muscle to target the genetic basis of disease. And it consists of three components. The first is the antigen binding fragment, or the antibody fragment, or the Fab, which is the top portion of this depiction of a full monoclonal antibody designed to bind to the transferrin receptor 1, or TFR1. The TFR1 is highly expressed across the surface of muscle cells, including heart, the breathing muscles like the diaphragm, and skeletal muscles. So the Fab targets transferrin receptor 1, or TFR1, and uses it to deliver the PMO to muscle while maintaining the ability of the TFR1 to transport iron into the cell. And that's its natural function. And I'll talk a little bit more about this in a moment. On the bottom part of the slide, you see this clinically validated linker. And we chose this because of its demonstrated efficacy and safety in third-party approved products. So what I mean by clinically validated linker is that this linker has been used in products that are on the market. They're products for cancer, but it's well understood. And so this helps us de-risk our overall novel therapeutic. And also this linker enables precise connection of the Fab to the exon skipping PMO. And finally, we have an exon skipping PMO, which we rationally selected to target the genetic basis of disease. This is where we leverage the modularity of the FORCE platform. And what I mean by that is for nuclear targeting diseases, such as Duchenne, we can choose a PMO, for example, and in this case, we chose an exon skipping PMO to target the specific mutation in the DMD gene. And DYNE-251 is our investigational medicine that uses an exon skipping PMO designed to enable the skipping of exon 51 to make a truncated functional dystrophin. The FORCE platform is designed with the goal of addressing the current limitations of delivery of PMO to muscle tissue, as I mentioned earlier, and really improve the delivery of PMO to cardiac and skeletal muscle, including diaphragm. And we're also looking to explore increased time between doses from those therapeutics that are currently on the market. And as we get into the clinical program, you'll see how that is. Next slide, please. So the mechanism of FORCE is different from other approaches. It's a question that we often receive. How is your mechanism, your modality different from other approaches? So we're essentially using the cell's natural biology to deliver our investigational medicine to muscle cells. Our Fab, as I mentioned, binds tightly to the transferrin receptor 1, or TFR1, and that TFR1 is highly expressed across the surface of muscle cells, such as those skeletal, cardiac, and smooth muscles. So the Fab acts as a kind of key that unlocks the TFR1 receptor and allows our PMO to enter skeletal, cardiac, and smooth muscle to target the genetic basis of disease at the level of the nucleus. In a way, we use a Trojan horse approach to drive the PMO into the cell using that TFR1 receptor without disrupting its normal function of transporting iron to the cells and maintaining iron homeostasis. So the FORCE platform harnesses the natural mechanism of TFR1 receptor-mediated delivery to transport the PMO across the cell membrane. And this is differentiating because we're not using membrane destabilizing agents to deliver the PMO into the cell or to leave the endosome. So you have this conjugate that enters the cell, you have the endosome that forms, the PMO is then cut or cleaved, leaves the endosome, and travels to the nucleus without disrupting the membrane. And by targeting muscle specifically, by using TFR1, we have the potential for a wide therapeutic index or a wide safety window, and also resulting in less frequent dosing and chronic administration. Next slide, please. So today, we feel Dyne is well on its way towards achieving the promise of force and delivering for patients. Over the last several months, we have provided updates on efficacy and safety, and today's update is going to recap the data that we released in May. And I'm really pleased to share this data in more detail. Next slide, please. So prior to starting our DYNE-251 clinical development program, we proactively obtained expert feedback. We engaged key opinion leaders, or KOLs, from various disciplines, including pediatric and adult neurology, cardiology, physical medicine and rehabilitation, pulmonology, and physical therapy. And importantly, we've gathered this feedback from across the globe. KOLs, both in the US and outside of the US, with experience running early and late stage clinical trials, have provided advice on our overall clinical development program, including the clinical trial design, the patient population that we selected, biomarker endpoints, functional endpoints, as well as key safety considerations. And importantly, they have provided key learnings from numerous long-term natural history studies to help inform our trial design and eventually contextualize the results we get from our clinical trial. One thing is very clear from the KOLs: they believe dystrophin continues to be a key endpoint for this condition, and we need to be able to restore dystrophin to be able to have functional impact. Importantly, we've also received advice directly from another group of experts, patients and caregivers, as well as global advocacy leaders such as PPMD, and from the Global Duchenne Community Advisory Board, comprising of advocacy leaders from several countries around the globe. They've provided insights based on their living with and being the voice of the Duchenne community, as well as experience with past and current trials. Now, this input has been critical in ensuring a patient-centric design, and it's included, but it's not limited to, key considerations for when families are choosing a clinical trial. Review of and input into our informed consent and assent forms, feedback on biopsies and functional outcomes, and overall really helping us identify the most burdensome or difficult parts of the trial, how to mitigate that burden to the extent we can, and finally, how to ensure patients and families are supported throughout the process. And we've taken this feedback and tried to make sure that we're addressing as much of it as possible, because we realize no matter what, being in clinical trials is still a significant burden. Next slide, please. So before I go into detail on the data, I just want to provide a very brief overview of the DELIVER study design. I know many of you have seen this in the past. Importantly, it's a phase 1/2 global trial that we've designed to be potentially registrational. The trial is enrolling ambulant and non-ambulant males with DMD who are ages 4 to 16 and have mutations amenable to exon 51 skipping therapy. Patients who have been previously treated with exon skipping therapies can enroll in DELIVER after a 12-week washout period. The primary endpoints are safety, tolerability, and change from baseline in dystrophin levels as measured by Western blot. And secondary endpoints include measures of muscle function like NSAA and time function tests, as well as we'll be looking at exon skipping. We've also tried to learn from recent data that has come out in recent studies. So we also have wearables now to allow collection of data such as stride velocity, which has been accepted by major European regulatory authorities even as a primary endpoint within Duchenne trials. For more details on inclusion and exclusion criteria, as well as the clinical trial sites in the US and outside of the US, you can visit the ct.gov page, which is listed at the bottom of the slide, as well as the EU clinical trials page. So those sites will give you much more information in terms of study design, inclusion, exclusion criteria, and the sites that we've selected so far. Next slide, please. Now, turning to some of the data, first of all, this slide goes through the baseline characteristics of the participants in the DELIVER study so far. As I mentioned, we have designed DELIVER to be potentially registrational. And so we want to be sure that we don't do anything to jeopardize the integrity of the study. So to maintain the blind, we haven't broken out these by placebo and DYNE-251 groups. Overall, baseline characteristics are similar across cohorts and generally representative of a population of boys living with DMD amenable to exon 51 skipping therapy. Next slide, please. So moving on to the safety profile, which we believe is favorable to date for DYNE-251 as of our data cutoff date, which is listed at the bottom of the slide. And based on 48 participants enrolled through the 40 mg per kg cohort as of that data cutoff date. We've not seen any dose-dependent increase in reported adverse events. Most of the adverse events were mild to moderate in intensity, and many of them were occurring around the time of infusion. Importantly, as of the data cutoff, no participants have demonstrated persistent related anemia or thrombocytopenia, kidney injury, or clinically meaningful changes in electrolytes, including magnesium. And no withdrawals have occurred either as of our data cutoff date. With approximately 480 doses administered over 35 patient years of follow-up, this has really supported the overall safety profile in our estimation and supported dosing up to 40 milligrams per kilogram. Next slide, please. So before I get to the data on the slide, let me just orient you to the structure of the slide, which is the same for the next few slides. The gray bar represents the placebo-treated patients. The blue bars are patients dosed at 5 milligrams per kilogram PMO equivalent dose, and the teal is 10 milligrams per kilogram PMO equivalent dose. And you can also see baseline and the change from baseline at the 6-month time period. And that 6-month time period is really important because it is the end of the placebo-controlled period. It is a 6-month placebo-controlled period. And then everybody switches to open label extension. Everybody switches to active drug after that 6-month placebo period. So this slide shows muscle concentration. And you can clearly see that DYNE-251 demonstrates dose-dependent increase in PMO muscle concentration. So this confirms that the drug is getting into the muscles and more gets in with higher doses. Next slide, please. This slide shows the exon skipping data. And here we also saw a dose-dependent increase in exon skipping. At the 10 milligram per kilogram dose of DYNE-251, this resulted in nearly 2% exon skipping. Next slide, please. And this slide is the dystrophin data. And we are particularly enthusiastic about this dystrophin data as measured by Western blot. At the 10 milligram per kilogram dose delivered every 4 weeks, DYNE-251 reached 3.2% normal dystrophin, a change of nearly 3% from baseline. This dystrophin analysis on the left-hand slide shows unadjusted dystrophin, and on the right-hand side, muscle-adjusted dystrophin, and I'll just go into that a little bit. So first of all, the dystrophin analysis is unadjusted for muscle content on the left side, and we believe this is an appropriate evaluation of dystrophin. And this data, in our estimation, in our opinion, is quite striking. However, we do know that others in the field have adjusted for muscle content, so we've done that analysis as well. And here you can clearly see how we've calculated this muscle-adjusted dystrophin. Basically, we've taken the dystrophin and divided by the percent muscle content. And when we look at this number through this calculation, DYNE-251 achieved 7.6% dystrophin at 6 months. So this is a change of about 7% from baseline with the 10 milligram per kilogram dose, dosed every 4 weeks. We do believe this is the highest level of dystrophin reported for an exon skipping modality. And importantly, again, this is at just the 10 milligrams per kilogram dose. And we're currently dosing at 4 times that level. So we're really excited to see what happens with higher doses. Next slide, please. So the 3.2% unadjusted dystrophin expression is nearing the goal of 5 to 10% unadjusted dystrophin, which we've said for some time that we believe will drive functional benefit, that will have functional impact. And what's exciting is that as we near that target level, we see dystrophin production lead to encouraging trends across multiple functional endpoints at just 6 months. We see some stabilization with the 10 milligram per kilogram of DYNE-251, while participants receiving placebo decline over that same period of time. And we see this impact through the NSAA, time to rise from floor, 10-meter walk run, and the stride velocity. So we'll continue to evaluate the functional impact at higher doses, as I mentioned, and at longer time points. And while we recognize that these are relatively small participant numbers, we're really encouraged by this data. Next slide, please. So in summary, we believe that these DELIVER data reinforce the opportunity for DYNE-251 and for the other exons that we have moving forward as well. And at the 10 milligram per kilogram dosed every 4 weeks, DYNE-251 showed a compelling profile in terms of the 3.2% dystrophin unadjusted and the 7.6 muscle content adjusted dystrophin expression. These trends and improvement in various functional outcomes like NSAA and time function tests, as well as a favorable safety profile as of the data cutoff date. Now, we do plan to provide an update on the path to registration by the end of the year. We know that several exon skipping therapies have been approved in the US, leveraging the accelerated approval pathway with dystrophin as a surrogate biomarker. We have confirmation that that pathway remains available to us, and we look forward to engaging global regulators even more in 2024. So we do believe the data set from DELIVER illustrate the promise of the FORCE platform in developing next generation exon skipping therapies, and we're excited about the opportunity to leverage FORCE to address additional mutations. We do have preclinical programs targeting additional exons such as 53, 45, and 44. We know there's a significant need for these for therapies targeting these exons, as well as other exons, and of course the rare exons as well. Next slide, please. So with that, I'd like to thank PPMD again for the opportunity to present to you today, and thank you to the families, investigators, the sites that have made this and so many other advancements in Duchenne possible with your countless sacrifices and incredible dedication. What you've all done over the years is nothing short of extraordinary. So thank you so much.
Thank you, Ash. So with that, we're going to move to the Q&A portion. So if you have any questions, please submit them to the chat function. And then we had some submitted ahead of time and some points that I think would be helpful for clarification. So if we can kind of go back to slide 5, and then I think there's a few of the slides we can kind of work through, and it'll be helpful to talk through some of those questions as we kind of go.
Okay.
Before we get that started, though, I know one of the questions that came in was from a family asking about, particular mutations of whether their child is amenable to exon 51 skipping. And for those of you who are maybe unsure, given kind of your genetic report, one, there are tools on PPMD's website. We have a, uh, deletion tool that you can kind of plug in your genetic variant and see if you're amenable to a exon skipping therapy. I think what's probably more helpful than that is there's a little kind of floating button on the bottom right hand of our website currently that you can schedule PPMD for you. So you can schedule a one-on-one with one of the many genetic counselors that work at PPMD, and they are more than happy to talk with you about your options, not just exon skipping in clinical trials, but other therapies as well. So just a good resource to have. All right, so wanted to start here. So talked a lot about TFR1, so the transferrin receptor. Just as to go over that again, can you just describe the different types of muscle where this receptor is found? Obviously skeletal muscle is important to function, but getting to smooth muscle to help some of the gastro issues, getting to the heart, these are very important targets as well.
Yeah, happy to. So, one of the things that Dyne was trying to identify early on was a target that would allow us to transport the exon skipping PMO to the muscles that are most relevant in Duchenne. And the the skeletal muscle, right? The muscles we use to raise our arms, to walk, etc. They This receptor is ubiquitous. It's highly expressed across these different kinds of skeletal muscles. We also knew that's not enough. That's not sufficient. We know that as the disease progresses, there are increasing pulmonary issues, increasing cardiac issues. And so we also needed to make sure we found a target that would allow us to be able to address the cardiac symptoms, the pulmonary symptoms. And again, the TFR1 receptor is highly expressed across these kinds of tissues. And I'll sort of get to maybe how we got there in a minute, but and then of course the smooth muscle. So we also know, while it's maybe talked about less, that there are GI issues, for example, in those suffering from Duchenne. So we wanted to make sure we could address the smooth muscle. So the jejunum, the duodenum, the gut, essentially. And it was really important for us to be able to also identify a target for those muscles. So it was a challenge to be able to get one target that is highly expressed across these key areas, while also making sure that target is not as highly expressed on other parts of the body, not as highly expressed in the liver and in the kidney. We wanted to make sure that target had as much benefit as possible with limited or no off-target effects. And so our scientists are phenomenal. And they came up with the TFR1 receptor that met all of that criteria of being highly expressed in those key muscles and expressed at a much, 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 much lower level in things like the filtering organs, the kidney, the liver, the spleen. So that target worked, very, very well. And there is a transferrin receptor 2, for example, which we don't target. So that target was seemed perfect. And then we went into the in vitro studies in cells, and then into the in vivo studies. So then we looked at the MDX mouse model. We published that data, and we validated our hypothesis in the MDX mouse model. And we looked at experiments, both exon skipping, dystrophin, as well as function in the mouse. And then we further validated this in a larger species, so in monkeys, where now there is no dystrophic model of monkeys. So what we can look at are two things, essentially. We can look at exon skipping in monkeys. So that was very helpful. And And we can look at distribution across various muscle and organ systems in monkeys. And again, our hypothesis panned out in monkeys where we saw this, ability to penetrate those key muscles with the TFR1 while managing any issues on the filtering organs. And so that target then is what we felt like, okay, we have the right target, and now let's do all of the right stuff in animals, talk to regulators, and then start this phase 1/2 study that we call DELIVER.
Great, thank you. That's a really helpful kind of breakdown. And then on this as well, so Ken, if you talked about the FORCE platform and just should DYNE-251 continue to have good safety and tolerability? Should there be good efficacy? Ideally, so you would be changing out, as you look at kind of the pipeline and other potential exons, it would be the PMO piece that you would swap out, and you'd maintain this linker in the Fab fragment that you're kind of evaluating currently, correct?
Yes, I think, yeah, thank you for bringing that up. I probably was not as clear as I could have been. So for our exon skipping agents, that is exactly right. The Fab and the linker will remain constant. It will be the PMO that we change. So here we have a PMO for 51 mutations, an exon skipping PMO for 51 amenable mutations. As we progress to 53, 45, it will be the exon skipping PMO that we will change out. So it's a highly modular platform that also continues to build on a safety profile and continues to get de-risked as we test more and more investigational medicines, both in the preclinical space as well as the clinical space. So we continue to build this bolus of safety information with respect to the platform as we go through our programs. And so it's a really good point where we're just exchanging that PMO.
Great. Thank you. If we go to the next slide. So two pieces here. One, I think just from maybe an educational standpoint, because we talk more, or we're having more conversations in the space as we hear companies coming with kind of, next gen exon skipping therapies, this being one of them, conjugating something to the PMO. So in the absence of that conjugation, how is the oligo normally getting to kind of muscle or other tissue, and maybe to help people understand kind of what the targeting purpose of this is?
Sure. Yeah. So a term that we use is sort of this unconjugated PMO or naked PMO. So in the United States, there are these unconjugated PMOs that are in the market, that are being used for various mutations, and they are delivered systemically, meaning intravenously. Our drug is also delivered intravenously, and so the drug then needs to get into the muscle cells, but it is really unclear how that PMO is getting into those muscle cells. Is there some sort of endocytosis that's going on, but how is that PMO getting into the muscle cells? And what we do know from literature is that much of the unconjugated PMO that is delivered through the veins is excreted. And so there is preclinical data out there, and there is other data out there that shows the inability of unconjugated PMO to get into muscle cells robustly in terms of skeletal muscle, but really not getting into cardiac muscle at all, and in a very limited way getting into diaphragm. And I think this is one of the reasons that these next generation exon skipping therapies are being evaluated. There is no doubt that this mechanism makes sense for this disease, an exon skipping PMO. It makes sense. It should affect the biology. It is really about delivery, about being able to get enough of that PMO into the muscle, into the nucleus of that muscle cell. So the next gen next-gen drugs are doing it in different ways. So there are companies that are using cell-penetrating peptides, for example, to be able to drive the PMO into the muscle cell, and that seems to be working more effectively than unconjugated or naked PMO. One of the challenges there is that again, in the clinic, there have been, for example, magnesium issues. So presumably it's getting into other organs like the kidney and stuff, where you want to try to limit leakage into the kidney. And then there are other companies, such as Dyne, that are using a receptor-mediated delivery to be able to drive the PMO into the nucleus of the muscle cell. So there are a lot of different modalities. Historically, the unconjugated just hasn't been able to penetrate the muscle cells effectively, and certainly not get into cardiac muscle to any great degree, especially when you look at the preclinical data that exists. And now you have cell-penetrating peptides. You have endosomal escape mechanism. You have receptor-mediated delivery. But all of these companies are doing amazing work, great work, to be able to solve the challenge of getting the exon skipping PMO into the muscle cells. So hopefully all of them work and are available to physicians and families.
Thanks, Ash. And then kind of along that line, so you talked a little bit about, the fact that you're, kind of co-opting some of the function of the receptor. So one of the questions that might come up from families is, is this going to have an impact on its normal kind of biological function? So have you seen any of that? How are you monitoring for that kind of potential reaction?
Yeah, it's a great question. So because we have, maybe I'll back up a little bit. So the TFR1 receptor, the nice thing is there's a lot known about it, about its natural function, to how it binds iron and transports iron and maintains iron homeostasis. So we use an antibody that is not competing for the same binding site as iron. So that helps mitigate disruption. Now, that said, we also have learned from the field there are many companies that are using the TFR1 to deliver different kinds of payloads for different conditions, including CNS. And so when we take the totality of information that is known in the public domain and what we've done in terms of the animal work, in terms of the biology of the receptor, in terms of what's happening in people, we make sure that we have the appropriate safeguards in our clinical trial design, in our clinical trial protocol. So we are very careful about monitoring, for example, iron, hematological parameters, anemia. All of those kinds of things are things that we are very acutely aware of, could be issues or could be disrupted with anything using the TFR1 receptor. So our protocol builds in the very rigorous monitoring for potential safety issues that could come up by using a TFR1 targeting agent.
Okay, great. If we could go to slide 9. So one of the things you mentioned here when you were just talking about kind of the phase 1/2 was considering or looking at it being a potentially registrational study. Can you just kind of clarify what you mean when you say that?
Sure. So for Duchenne, as I'm sure most people know, the exon skipping drugs that have been approved under an accelerated approval pathway by the FDA in the US. And in short, this pathway is a phenomenal pathway for families, physicians, for the community, because it allows an approval to be based on a surrogate biomarker, that is likely to predict clinical benefit. And the FDA has opened up a pathway whereby modality or a therapeutic agent that shows there's an improvement in dystrophin restoration can get approval through this accelerated approval pathway because dystrophin, a change in dystrophin, is likely to predict clinical benefit. And the other benefit around, the other nice feature of this is that instead of waiting for 5 or 6 years for a study to read out to show a functional difference between a group that's treated versus placebo, or a group that's treated versus natural history, is that this allows for a much more expedited approval. And this can happen while a company is continuing a investigation for confirming there is functional benefit. And that would be a requirement for any company that, yes, if they get approval through the accelerated approval pathway, they must have a confirmatory study that shows and confirms that there is actual clinical functional benefit of the drug.
Thanks for the clarification. I think that's helpful. Just always want to make sure anytime we start to talk regulatory, everyone kind of has an understanding of what we're discussing. And then I think there was a question submitted here that I think can kind of go along with this. So maybe the first part of it is, so someone's asking about combination therapy. So I think first maybe talking about within the kind of confines of the study, what other therapies are or aren't allowed. So you already discussed that with the exon skipping therapies, whether investigational, I believe, or approved, there, as long as they wash out, they can participate.
So that's right. So yeah, exactly. So I would remind everybody this is a phase 1/2 safety tolerability study where we are also looking at dystrophin. And because it is our first in human study, we need it to be a bit more controlled to ensure that we are first and foremost confirming safety, identifying the optimal dose and dose regimen, and being able to show some dystrophin restoration as well, which I described earlier in the slides. So I think that is really important context. So we do limit, first of all, anyone who has been on gene therapy in this phase 1/2 study cannot enter the study, because we really need to understand what DYNE-251 can do largely on its own. We do allow those who have been on other investigational medicines to enter the study, as Eric said, with a washout period, so with a 12-week washout period. And so there are criteria that limit folks to come into this study unless they meet some of these parameters if they are on other drugs.
And then with the other approved therapies, so our more recent approvals of Agamree and then Duvizat, are patients able to be on those therapies and participate in this study?
Yeah. So for vamorolone, yes, we do allow it. For givinostat, currently we are not allowing it in this particular study. I think it's something that we are carefully considering. Again, because this is a phase 1/2 study and it has to be, a bit more restricted, a bit more homogeneous. It's a little more difficult to get a clean read on our treatment effect if we included a drug like givinostat. But we are having active conversations about how we manage this and other modalities that are emerging and likely to be approved.
Thanks. And I understand, and that's helpful for the within the study design and having something really tightly controlled so you can show, efficacy and evidence really well. Their question was around the combination piece. So if DYNE-251 was, so just taking it a step further, so I did want to start with inclusion, exclusion criteria. The next piece of that is, if this were to be approved, theoretically, would something like DYNE-251 and, givinostat, Duvizat, would those be, therapies we could, think about combining?
So yeah, I want to be careful here. I think that again, I'll always say please talk to your physician and other inform parties about these kinds of considerations. Though I have many thoughts and opinions around this, I think, what I can say is there is no data, right, to be able to point to a clear, definitive, yes, you can combine DYNE-251 with another one of these therapies that are, approved, like Givinostat. So there, I can't say yes, this is something that should be done. You need to talk to your physician about this. That said, um, in general, I would say that to have the best outcomes, there are going to be combination treatments necessary in in Duchenne. So not even speaking about DYNE-251, but thinking about exon skipping drugs plus HDAC inhibitors, or gene therapy plus this, or I think combination therapy is something that as a community we need to be thinking about, we need to figure out, and frankly, we need to generate some data so that we minimize any sort of we don't miss opportunities, and also so we don't do any harm.
And to the person who asked that question, and just to echo kind of what you said there, Ash, I mean, PPMD feels very strongly that combination therapy is something that we are going to need, and we want to ensure all of our families have access to therapies that would benefit them. But along with that, along with not just the access piece, is for us to make sure that we are collecting data so we can understand what is working and what isn't working, what is safe, and how best to proceed. But that is something that I think we all, or a lot of us, acknowledge is going to be a really important step for us moving forward.
Yeah. And may I pause for
Yeah.
1 second? Sorry. I know, Catherine and I have talked about, kind of the treatment paradigm shifting over time, and actually it's here. So maybe I'd invite Catherine, is there anything else you'd want to add around sort of the shift? Because you've been in this space, frankly, longer than I have, and you've seen more than I have.
No, I have very little to add. I think the idea of combination therapy makes sense. This is, a condition that is multi-systemic. Treatment approaches may be targeted, right? Which a priori means you have to go after multiple targets, multiple drugs, things like that. And a kind reminder to everyone that I think that state of combination therapy is already present, right? I mean, it's steroids with the backdrop of everything else that is added, right? So it's what is the data that we have there, and what is the data, to your point, Ash, that we continue to accumulate as trials advance and additional medicines come on the market.
And then kind of staying on this slide, we had a few questions around doses. So one is, so you're shown data here for the 5 and the 10 mg, going all the way up to 40. I guess what are the other doses that you're doing in the trial? And then as you continue to enroll patients, are you enrolling at all of the doses? Have some of them been dropped and just moving to some of the higher doses? Can you just talk about that?
Sure. Yeah. So again, if you go to our corporate deck, you'll see more detailed slide, but we are dosing up to 40 milligrams per kilogram. And that is, per the DELIVER protocol, that's the highest dose as per the protocol.
Okay. And then in the open label extension, you said, for those who are on placebo, they will be moved into dosing. Is that a fixed dose that everyone is going to move on to? Is it kind of their current dose, and then as you get additional data, you'll make a decision? So can you just talk through kind of what that kind of dose is in the open label extension?
Yes, for sure. Yeah. So sorry, it's a great question. So the way that we've designed the study, which I think is good for patients, is that we're able to escalate to the highest safe dose within the study. So after the 24-week placebo-controlled period, everybody enters the open label extension, gets active drug, and then gets escalated to the highest safe dose. So, if you are on 2.8, again, this is theoretical. If you are on 2.8 and the highest safe dose is deemed at 10, then you can jump right from the 2.8 mg per kg at the end of that placebo control period up to 10. So that's the way it's designed.
Good, thanks. I think that's helpful for family to understand. So I think there's always concern anytime families enroll in studies where there's potential placebo is, am I going to be on placebo? And then once the open label, what does that look like? So that's really helpful. If we go to slide 11, I think there was just one thing you said here that I want to clarify just to make sure you can talk about so families understand. So I think you said maybe somewhere over 400 doses have been administered to this point. You can correct me if I had that number incorrect. But you said so 35 patient years. Just when you of kind of data, can you explain what that means?
Oh, yeah, I think it's an interesting term. It just tries to sort of so it's known as a person year, and I guess essentially it's a unit of measurement that we use in clinical studies and statistical jargon as a way to measure the amount of time a person contributes data to the study. So it takes into account the number of people in the study and how long each person is involved. So, if you have a study that follows 1,000 people for 1 year, it would have 1,000 patient years of data. I hope that helps.
Yeah, it does. I think that's just one of those, to that point, it is one of the jargon terms, and I always just want to make sure we talk about it, because if someone hears that or they kind of mishear and they think, okay, like how many years did they say? Just to help clarify so know that it's appreciated.
Yeah, it's essentially the number of patients times the number of years they've been. Yeah, that's yeah.
And then on slide 14, so one of the things you talked about here, and I think we've seen in some other presentations, is some of this muscle content adjusted dystrophin. So I think just talking through a little bit of the rationale. So I think everyone probably on listening in has an understanding that as the patient's age, as the muscle tissue kind of undergoes repair and replacement, you eventually have some of the muscle being replaced with fat and fibrosis. So can you just explain how are you actually getting kind of to this muscle content adjusted dystrophin? So you're taking one of the biopsies and then doing, how are you getting to that?
And then evaluating essentially evaluating the fibrotic content of that.
So the functional muscle versus the fibrotic content in that slice of muscle.
Yeah, exactly.
I just wanted to talk about it because I think we're starting to see this more, and so it's helpful for everyone to understand when they see that those kind of two different numbers, because it can be hard sometimes to interpret. I think if you're a family and you see this and you say, okay, well, I'm seeing two different numbers for dystrophin, what do they mean? So that's helpful.
And I think this is really important. So the unadjusted dystrophin, as I mentioned, is what we anchor on, because we do believe, as I've said in the past, that 5 to 10% unadjusted dystrophin is going to hopefully drive functional impact. And we as I said, we already started to see it at 10 mg per kg. Now, the muscle content adjusted dystrophin, I think, helps provide an additional piece of information. And when we've talked to key opinion leaders about this, they have appreciated that we're putting both numbers out there transparently, how we're calculating it, and just so it provides additional context, when they do look at our data and data from other academic institutions or other sponsors that are doing drug trials. So I think this is just a way for us to be as transparent as possible. And I do think that there's more work for us to do as a community, especially sponsors with partnership with PPMD and others, which is to be, put our methodology out there very transparently, and be able to share across all of our sponsors with you how we're doing this in a detailed way, so that we can avoid the very thing that you just talked about, which is there are many sponsors with different numbers out there, have done things differently. And I would imagine it's extremely difficult for families to truly interpret what they're seeing from from different studies.
Thanks for that, Ash. And then if we go to slide 15. So first of all, I do want to say thank you for acknowledging when you were describing this, this is a small number of patients. I think that's always important and relevant for families as we kind of start to look at data that's coming out. But, I do think it's also important to highlight some of the, stability that we're seeing across here. That was something that came up at PPMD's advocacy conference in some of the discussions we had with family in terms of discussing, kind of benefit from therapies and what's important, to individuals. So this is also good, as you discussed, the surrogate piece of, the approval pathway and the likelihood of increased dystrophin to provide benefit, but having some functional data to go along with that, I know is helpful for families as we kind of evaluate this and as they make decisions, because they have a lot of options. So it's always, it's helpful. We did have a question come in, so this is for the 10 mg per kg on, timelines for when you might expect to see more data from those on the kind of those higher dose ranges that you were discussing.
Yeah. So I'll probably be a little unsatisfying here in terms of my answer. So, we haven't committed to when we'll disclose the next set of data. As I said, not only from higher doses, but also longer term data, which we think is also really important. So as we are ready to communicate timing for the next data release, I'll be running to you guys and letting you know. But for now, we haven't disclosed an update in any level of specificity as to the next data disclosure. But we're excited about that next set of data that we'll be disclosing. We're enthusiastic about what we're going to see, and we don't know what it is, but we're eager to see it as much as you are. So stay tuned, and hopefully I can come back to you with an answer to that.
Thanks. And I do want to note, they did also just want to acknowledge and say they were very happy with the support they've received from Dyne throughout their participation in the study. So always good to hear feedback from participants. And then, so with that though, for the functional data and or other data that you're collecting, as patients move into the open label and the long-term extension, will there be additional data collection there, or is the functional going to be just kind of the 6-month marker for all patients in the study?
So sorry, there was a little blip. Can you repeat your question?
Yeah, just what is there additional data that will be collected during the open label and the extension study? And I was just trying to get at, is that going to be functional, safety data, kind of what you might be collecting?
Yeah. So the endpoints are the endpoints that we're collecting in the 24-week placebo control period. So for example, these are just a few of them, but we'll be collecting the same endpoints. But yes, we will be collecting functional data and safety data over the entire duration of that study. So in totality, it's a 3-year study. Now, the frequency of data collection may shift in that long-term extension component. So we have the 24-week placebo, the 24-week OLE, and the 96-week long-term extension. And so to manage the burden on families for that long period of time, we will have, for example, less frequent collection of functional data. So we will use the same endpoints, collect them as frequently as we are now for that kind of first year, and then go to a less frequent data collection for years 2 and 3.
Okay, great. And then the last thing is if we could go to that final slide, I just wanted to make sure that, so there's Catherine's, I think the one after this, contact information is there. So that way, if there's additional questions from any families on the line or anyone watching the recording, you can reach out to Catherine. And then finally, if families are interested in participating as you continue enrolling for this study, just what's the best place for them to go to make outreach if they're interested in enrolling their child?
Sure. So Catherine, I don't know if you want to take that, but reaching out to Catherine. Go ahead, Catherine.
Yeah, no, there's a couple different routes, right? You can certainly feel free to contact me, and I can provide that information as well as direct them to clinicaltrials.gov here in the US and the equivalent in the EU, or they can access those links directly, which were a little bit earlier in the presentation. I know it's not ideal, but it is where the most comprehensive trial information can be found. And if folks want to access that and then follow up with questions, always free to reach out to me directly.
Thanks, Catherine.
Thanks, Eric.
Yeah.
24 hours a day, 7 days a week.
Yeah.
You're right. Yeah.
3 a.m. Contact Catherine.
I'm on it.
Well, I just wanted to say thank you for joining us for this webinar. I do really appreciate, as we kind of go through those questions, Ash, you taking the time to kind of really dive into some of them. I think that helps, just as we kind of, all of us continue our education on this and learn more and help, all the patients and the families feel empowered in decision making. That it's helpful to kind of go through all of that. So I do appreciate that.
My pleasure.
And so again, for anyone, if you have friends or family who this would be of value to, we will have this recording up on PPMD's website within the next week. If you have additional questions that didn't get answered or that you think of later, please feel free to reach out to PPMD or to Catherine, and we'll make sure that those get answered. So thanks everyone for joining today. Thank you again to the Dyne team for being with us. I hope everyone has a good afternoon.
Thank you so much,
Eric,
I
Emily.
appreciate it.
Yeah.